Good morning, everyone. Welcome to the Theory and Methods Contributed Talk session. And I'm Zelin Yu. I'll be the host of this session. And thank you all for being here and joining us. I encourage you to save all the questions until the end of each talk. And if you have any questions, please come to the front, there are mics on both sides. So, yeah, with that, let's begin our first presentation, a non-local variational framework for optimal neural representations by Jon Tian. Please join me in welcoming Jon.
Thank you for the introduction, and thanks to the organizer for having me. And this is joint work with Professor Brent Dora at the University of Chicago. We're interested in the discriminability problem for one-dimensional circular variables, such as shifting gradient orientation, head direction, etc., which we denote with the variable theta. Shown on the left is a common experimental setup where the animal is asked to distinguish between different orientations, here theta 1 and theta 2. Suppose that we have a population of n neurons with response denoted by the vector x, such as by count vector, that encode this variable. So shown on the right is a schematic of the neural response distribution to theta 1 and theta 2, denoted by the two colored point clouds. Then the overlap between these point clouds is assumed to be the source of the error. Then the goal of the code is to arrange the family, one-dimensional family of point clouds such that the overlap between the distributions are minimized. Formalized mathematically, this says that we want to find a map f from theta to p of x given theta such that the cost function L is minimized. This problem has been studied for decades, but previous work has a major caveat, which is they often use fish information as the measure for the quality of the code, but it is a fundamentally local measure. To see what this means, let us look at this toy example, where from left to right we have three population codes with the same fish information but decreasing mutual information. This is because fish information is only sensitive to ambiguities locally, which denoted here by the blue arrow, but it cannot detect global jump errors, denoted here by the green arrow. We can also see this in the neurometric curve, where here is classification error plotted against the delta theta between the two thetas being distinguished. We see that in the fine discrimination regime with small delta theta, the three codes have virtually the same performance. But in the coarse discriminant regime, they have very different performances. So we want to find a cost function that captures both the fine and coarse discrimination. And our proposal is very simple. We simply integrate over delta theta to get the cost function on the left, which is the average binary classification error between all pairs of thetas. Because we are integrating over all pairs, this is a global measure of the quality of the code. If we make certain assumptions about the noise structure, such as independent Poisson, then we can establish a bijection between the average response and the mean response and the full distribution P of x. This bijection allows us to replace the map F with the map R, which is exactly the tuning curve. Then our goal becomes optimizing over all tuning curves. This is also a classical problem, but previous work often assumes some kind of functional form for the tuning curve, such as Gaussian bumps, and with a finite number of parameters, such as width, height, and density. This makes the optimization problem more tractable, but a lot of these assumptions are not justified. But here, we don't make any assumptions on the functional form, and any smooth tuning curve is fair game. So this makes our problem a non-local variational problem. So in this work, we analytically derived the solution to this problem, but in the interest of time, I will not go through all the derivation, but only give you a sketch of the ideas. Going back to the bijection between the mean response R and the full distribution P of x due to assumptions such as independent Poisson, this says that the space of neural response distributions is an n-dimensional manifold, and we can put a Riemannian metric on the manifold according to information geometry. And this gives us an n-dimensional Riemannian manifold M of neural response distributions. And we call the geodesic distance on M D, which is the Fisher-Rao distance. And previous work have shown that the classification error L is approximately a function h, shown here on the right, of the Fisher-Rao distance D. And luckily, for a lot of noise models, the manifold is Euclidean. And we can make this structure explicit by doing a coordinate transform from the finite space R to an alternative coordinate y, where the Fisher-Rao distance is explicitly Euclidean. This allows us to invoke a result from knot energy theory to conclude that the minimizer of the cost function is the round circle in the y coordinates. So shown here in this schematic, on the left is the finite space, and on the right is the Euclidean coordinates, y coordinates. To get back the optimal tuning curve, we can simply invert this transformation to get back to the tuning curves. And for Tian Poisson, this transformation is the ln y square. So we call the resulting code the square of circle code. We want to point out that this transformation depends on the noise model you impose on the system. And different noise models will correspond to different coordinate transforms. Here's a visualization of what the code looks like. Compared to classical codes like the von Mises code on the left, our square of circle code exhibits a wide range of heterogeneity across neurons. And in terms of performance, we validated that indeed our square of circle code outperforms the classical von Mises code in terms of coarse estimation. We validated several predictions in head direction cell dataset shown here. The first prediction concerns the shape of the tuning curve. Because each tuning curve is a U-shaped function of the cosine, we predict that first there should exist some neurons that have bimodal tuning curves, and that these bimodal tuning curves have phase difference between the peaks that is pi, exactly pi. And this is indeed the case in the neural data. The second prediction concerns the relationship between variability and tuning shape. So recall that we said that the transformation between the L coordinates and R coordinates depend on the noise model. Then a different noise model should give us a different transform than the square. To validate this, we consider the super Poisson model of the noise, where the fan rate of each neuron's Poisson rate is modulated multiplicatively by a variable g, which is a gamma variable with mean 1 and variance sigma squared. Under this model, the variance is a quadratic function of the mean, with larger sigma giving you more super Poisson variability, shown on the right. And under this new noise model, the optimal solution is this new transformation of the circle. And intuitively, the effect of larger sigma becomes sharpening the tuning curve. So these two sigmas are statistically unrelated quantities of the neuroresponse distribution. And we can independently fit two sigmas from the two aspects of the neuroresponse. But our theory predicts that these two statistically unrelated variables should be correlated with each other. And this is indeed the case in the neural data. So what this says is that the more super Poisson a neuron is, the sharper its tuning curve should be. So here are some discussion points. First, we define a canonical geometry in the space of neuroresponse distributions based on neural variability. And this turns a statistical question into geometric ones. For example, here we are seeking the optimal tuning curve for the optimal neural response distribution for a discriminability. And this phrased in a geometric way, this says we just need to look for circles in the appropriate coordinate system. And finally, this says that noise is isotropic and the statistical distance is Euclidean in these y coordinates. So this suggests that this may be a better space to look at neural response representation neural representation in. For example, during representational similarity analysis, where the calculation of the representational similarity is implicitly based on the assumption of Euclidean space. And this may be better carried out in the space where we have a theoretical reason to believe that there is a natural statistical distance that is Euclidean. With that, I'd like to thank the Doron Group and as well as the Grossman Center at the University of Chicago. And if you're interested, please come check out my poster, D1, in the afternoon. Thank you.
Since you've successfully verified the predictions with the in vivo dataset with mice, have you, or how well do you expect the framework to be generalized to other species or even other recording modalities, like not in vivo? Or, yeah.
So we actually did look at a V1 dataset in monkeys, but it's just not shown here. And we did see good validation of the predictions. And yeah, so I think the key here is that as long as you have this noise model with correspondence between mean and the distribution, then I expect there to be some validation there, yeah.
Okay. Thank you so much. And thank you again. And our next speaker is Yuqi Zhang, and who will present Hyperalignment reveals shared information in monkeys' idiosyncratic fine-grained movie fMRI patterns. Please join me in welcoming Yuqi.
Okay, thanks for the introduction. And this is a joint work with Professor Wim Spandov's group in KU Leuven. And today I want to show you that the macaque's brain share a high-dimensional representational space that anatomical alignment alone cannot reveal. And I will walk through the 3 analyses we do to test it. So why do we study monkeys? We can do more tests in monkey than in human. And in macaque, we know that the information carried by patterns of population activity, we can decode it within regions such as the face and body patches. We can line up the coarse structure across brains anatomically, but we cannot do that in a fine-grained structure because every brain is different. That's exactly why hyperalignment was built to do. Instead of lining up the brain anatomically, it projects each brain to a shared model space learned from the response patterns, usually during a naturalistic movie viewing, or from functional patterns, functional connectivity patterns. It works well in human, but nobody has ever applied it to monkey data. So that's our question today. Does hyperalignment reveal shared structure in macaque's idiosyncratic fine-grained topographies? To answer this question, we first collect a data set at KU Leuven, while 12 rhesus monkeys were scanned at a 3T scanner with head fix and free viewing. They watched a movie called Monkey Kingdom because we think it will make the monkey more engaged if they're seeing their own kind. And the movie was split into 5 clips of 15 minutes each. And each monkey saw the movie about 8 times. And then we average across those repeats, which gives us a higher signal-to-noise response time series. And the core idea of hyperalignment is that it aligns the brain activity across individuals by rotating each subject's unique neuron patterns into a shared model space while preserving their underlying information structure. Given the data set we have, we are using response hyperalignment to align movie response to a common model space. So here's how we build and test the model. We first use the first 3 clips of the movie from all 12 monkeys to compute a whole brain template. Then we estimate a transformation matrix for each monkey using ridge regression. Everything afterwards is the validation. For the first 2 analyses, we apply those transforms to the 2 held-out clips and then hold 1 monkey out and use the rest 11 monkeys, comparing to the other average for a correlation or predicting it from the others for movie time point in decoding. The third analysis is a bit separate because it used different transforms. For the 4 monkeys with localizer data, we estimated pairwise transforms from all 5 clips to predict each monkey's own functional topography from the other 3. So here's the result of the first test. It's the simplest version of the question. Do different monkeys respond the same way at the same moments of the movie? For every vertex, we correlate 1 monkey's response time series with the average of the others. Mean to subject correlation nearly doubled from 0.253 with anatomical alignment to 0.496 after hyperalignment. And every one of the 12 monkeys improved, so it isn't driven by a few subjects. The larger gain were in occipital, temporal, and lateral prefrontal cortex, which means the improvement is not confined to visual areas. So we are aligning higher order cortex too, not just low-level response to the movie. But higher correlation only tells us response line up. Does what line up actually carry information? That's our next test. Using only other monkeys' responses, can we identify which moment of the movie a given monkey was watching? We run 2 separate analyses, one at the whole brain level and one at the searchlight level. Both analyses use the same cross-validation method. We hold 1 monkey out and use the other 11. For the whole brain, we first reduce the dimensionality with PCA, then pick the number of components from the training data and match each held-out time point to the nearest response pattern in the other monkeys. The identical analysis run inside nearly 20,000 small searchlights with the accuracy assigned to the center vertex, so we can see where the shared information lives. For the whole brain, the accuracy went up from 0.24 to 0.43, roughly an 80% of increase. There are 900 time points, so the chance level is about 0.1%. Both anatomical align and hyperaligned accuracies are far above chance, but hyperalignment recovers substantially more. Now look at the curve on the right. It takes more than 70 principal components to reach peak accuracy. and hyperaligned data beat anatomical aligned data at every number of components. So whole brain patterns tells us the information is there, and the searchlight tells us where it lives. So when we restrict the analysis to local patterns inside small searchlights, mean accuracy goes from 0.005 to 0.028, more than a 5-fold increase, with the highest accuracy in posterior inferior temporal cortex. Notice that the gain is proportionally much larger locally than it was for the whole brain. That's the inference that hyperalignment is aligning generally fine-grained patterns, not just the coarse regional signal that anatomically could already capture. And the strongest shared representations say exactly what you'd expect from a naturalistic movie, inferior temporal and lateral prefrontal cortex. So everything so far is about what monkey shares. The last analysis asked the opposite question. Does the model also capture what makes each subject different from the others? So only 4 of our monkeys have face and body selective localizer data. For each one, we estimate a transform from each of the other 3 using all 5 movie clips, and then we apply the transforms to align their localizer map into the target brain as a predicted map. And then we average the 3 predictions and compare that with the target monkey's own measured map. And here you are seeing a sample from the 2 monkeys, M3 and M4. And the left is each monkey's measured map, and on the right is monkey's predicted map from the other 3. And as we heard from Doris Chow's talk, the monkey's brain has specialized face patches like ML, MF, and AL marked there. And the prediction actually recovered those face patches. Comparing M3 and M4 to each other, their measured maps differ, and the predicted map also differ in the same way. So transforms estimated purely from the movie watching carry enough information to reconstruct an individual subject's category selectivity topography from other brains, including the parts that are specific to this subject. So to summarize, macaque's cortex holds a high-dimensional shared representational space that anatomical align cannot reveal. And hyperalignment recovers both what monkeys share and what makes each animal distinctive. And this 3 task goes from a single vertex level to a fine-grained response pattern, and then to the core-scale functional maps. And at the end, I want to thank for my lab and for all my collaborators, and for all the people who collect the data. And I have a poster today at D2 in the afternoon. If you have any questions and want to talk to me or have some suggestions, welcome to come talk to me. And there's 2 other posters from my lab too if you're interested. And one of my collaborators is recruiting, so feel free to reach out. Thank you so much.
Thank you, Yuqi. It's a nice talk. And we'll be able to take 1 to 2 questions from the audience. Anyone? Okay, so I have a question just out of curiosity, because I noticed that in your extended abstract, you mentioned like you applied this framework to both monkeys and humans.
Yes, due to the time limit, I'm only showing monkey here, but human shows like consistent result, and cross-species work is in progress.
Oh yeah, that's really interesting, because I just want to ask, like, did you extend this hyperalignment across species?
Yes,
like
we're doing that right now. Yeah, so hopefully we can see some results soon.
Yeah, that's exciting. And thank you again for sharing with us.
Thanks.
And, yeah. So let's welcome our next speaker, Angeliki Papathanasiou, who will present her talk, Task-Guided Cross-Subject Latent Alignment, a Multi-Encoder-Decoder VAE. Thank you.
Hi, everyone. I'm going to be talking on our work on cross-subject latent alignment. Individual brains vary substantially in terms of their anatomy and their functional organization. Cross-subject alignment aims at transforming individual neural spaces into a shared common space. This is finding the projection transformations that recover a common space where all subjects can be projected and aligned. This allows us to uncover commonalities and differences across the subjects and enables population-level analysis in this common space. Traditional methods like shared response model and procrustes, which are the methods that we're comparing with in this work, require shared stimuli. And they learn the alignment, namely the projection transformation to this common space from the neural responses to this common stimuli. Usually, for naturalistic paradigms, subjects view largely non-overlapping stimuli, so deriving the transformations from this small subset can result to overfitting. And also, there are cases where this one-to-one stimulus correspondence between subjects cannot be controlled. For example, interactive environments, video games, etc. We worked with the natural scene dataset that is comprised of the fMRI data of 8 subjects that view mainly non-overlapping stimuli. Each subject views 10K images, out of which only 1,000 are shared between subjects. To overpass this need for common stimuli, we propose a method that uses artificial neural network representations to the same images that the subjects view as an alignment scaffold. So given a dataset that has a very small overlap like NSD or even not at all, we can feed all these different stimuli to an artificial neural network. Here we take a ResNet and get the representation for each of these stimuli. So basically for each image we have the non-overlapping fMRI activities of 8 subjects, and we have the full coverage of these corresponding to ResNet activations. So basically we will try to align each subject to the ResNet instead of each other, and then implicitly achieve alignment, cross-subject alignment. Here's the architecture that we're proposing. It's a multi-encoder, multi-decoder, variational autoencoder architecture. We have subject-specific encoders that take the neural responses of subjects and project them into a common space. This is where we're going to have this cross-subject alignment. We have a ResNet encoder doing the same for the ResNet activations, and we have subject-specific decoders and a ResNet decoder. So all these of 4 pathways are active at the same time, and we train them at the same time. If we are to focus on the fMRI towards the ResNet pathway, this is the primary alignment mechanism, because if we think the subjects are trying to project their responses into the common space, and all of them are going to be trying to reconstruct the same computational target, ResNet activations. So basically, if we think of 2 subjects viewing similar images, let's say 2 different images of a dog, the fact that they will be reconstructing the same, not the same, but like very similar ResNet activations imposes them to be close in the latent space, and thus we implicitly achieve this alignment with each other. After the training, and when we have achieved the cross-subject alignment in the latent space, the ResNet encoder and decoder are not anymore needed for the inference. Here we visualize the latent space, where with different colors we see the different categories of the NSD, for example, food, animal, etc. And we see how our method on the left achieves superior semantic organization. We also run 2 controls, one with an untrained ResNet instead of the ImageNet pre-trained one that we use in our baseline, and one where we have shuffled the one-to-one correspondence between the ResNet and the fMRI data. And these 2 controls show that this nice semantic organization that we see here requires an ANN backbone and also task-relevant representations. We also explore a more fine-grained scale metric where we take the latent representation in the common space for one subject's stimuli, and we take the k nearest neighbors from another subject's representations, and we look if the same stimulus is included in them. Our method outperforms the other methods. For example, here in 46% of the cases, the closest neighbor to one subject's latent representation is another subject's latent representation for the identical stimuli. So we have achieved stimulus level discrimination. The same stimulus maps consistently to nearby regions across subjects. Staying in this common space, we quantified the cross-subject alignment with a component correlation that quantifies if different latent dimensions of the common space encode similar things across subjects, for example, animalness, and with RSA, and we see how our method achieves higher alignment in this common space. In the interest of time, I'm not going to show these results, but we have them in the paper. It is important to make sure that enough information is preserved in the common space to reconstruct back to the original subject spaces, and we have shown that all of the methods have equal preservation of stimulus-related signal. This is important because this shows that this higher alignment and this nice categorical structure that we get in the latent space does not come to the expense of reconstruction, of preservation of stimulus-related signal. In order to see if the common space captures a shared neural structure that generalizes across individuals, we take one subject's latent representation and predict another subject's neural activity. Here are the results showing that for all possible pairs, our method outperforms the compared ones. So to see if this cross-subject prediction that we saw before preserves sufficient semantic content to drive a complex task, we take the result of subject 2, let's say here, neural prediction, and feed it to a pre-trained frozen image reconstruction model and derive subject 1's stimulus. Here are some image reconstruction metrics for this cross-subject image reconstruction and some actual visualizations that show that we get better reconstruction with our method and also more consistent across the different subject pairs. So to sum up, our method removes the search stimuli bottleneck and recovers a common space with improved semantic organization, higher cross-subject alignment at no cost of the preservation of stimulus-related signal, and we also showed improved cross-subject neural prediction, also showcased with image decoding. Future directions involve to actually take this common space and do population-level analysis there, and extend this to other brain areas, because here it's only visual cortex. Thank you. And here's the QR for the full paper.
Thank you, Angeliki. Is there any questions from the audience? I have a question. That is, your framework uses residual network 50 as the scaffold, and I wonder, does the learned alignment preserve the hierarchical order of the visual cortex, of the biological visual cortex, or it just learned the representation as a single global representation?
So the ResNet here works as an alignment scaffold. So you can do this with many different other models, and we have in the paper also in the appendix we have like some other comparisons. ResNet, though, because it resembles and it has this hierarchical structure, at least with the models that we have compared, performs better, and it could be because of exactly what you're saying.
So this might be because it preserves some kind of hierarchy.
Yeah, so this is also very interesting to see why the different models can result in different alignment levels, and this is maybe also can be used to even compare models with brain data as like their representation alignment, basically.
Thank you so much,
Thank you.
That's a really great talk. Thank you again. And so let's welcome our next speakers, Navve Wasserman and Roman Belli. They will present The Wisdom of a Crowd of Brains, a Universal Brain Encoder. Please join me in welcoming Navve and Roman.
Hey, my name is Navve, and together with Roman, we will present our work, The Wisdom of a Crowd of Brains, a Universal Brain Encoder. So let's start. When a person sees images, what happens in their brain? We know there are, for example, can we learn what is encoded where in the brain? We know there are brain areas that correspond for specific concepts. What other exists? Functional MRI, as you know, is a brain imaging technique that may help us answer those kind of questions. However, since data acquisition is expensive, there is not enough data to span the huge space of brain functionality. So can we simulate brain activation for many more images? Given images and corresponding brain activity, we can train an image to fMRI encoder that learns to predict brain activity from an image. Then we can take many unseen images, predict their corresponding brain activity, and use that for having much more data to explore the brain. However, for having a good encoder, we need a lot of training data, which is still unavailable. So what can we do about it? For example, can we combine data for multiple subjects? But nothing is so simple. Why? Because our brains are not the same. Given two individuals seeing exactly the same image, their corresponding brain activity is not the same. Therefore, most existing encoders are trained separately per each individual. So, can we efficiently combine data from multiple brains? We show here that the answer is yes. We present a single universal encoder, trained jointly on different subjects, dataset, and fMRI machines. Our method combined datasets collected containing many individuals who saw completely different images and were even scanned on different fMRI resolution machines. This allows us to integrate datasets collected over the years and across the globe. This provides us much more data and therefore much better encoders, but also a way to discover shared functional regions across brain, as you will see later. So for that, we propose to use a voxel-centric approach. We have many individuals with different fMRI resolution. Each individual has different number of brain voxels. Each brain voxel and each individual will be assigned with a voxel embedding vector. Those vectors are initialized randomly and will be later optimized to represent each voxel functionality. This optimization allows to learn a shared voxel embedding space, a space where voxels with similar functionality will be closer together. So how do we optimize this voxel embedding and the network in general? We have an image, and we want to predict a voxel activation and respond to this specific image. We first extract image features with image foundation model, both low and high-level image features. Then we will use the assigned voxel embedding to this specific voxel. We want to allow this voxel embedding to attend to the specific image features that correspond with its functionality. For that, we are training a voxel image cross-attention that integrates the information to predict the voxel activation. Importantly, the voxel embedding is voxel-specific and not image-specific. The voxel embedding is used to predict its voxel activation for any image. Therefore, it must learn to encode what the voxel cares about in the image. For example, whether it cares about low or high-level image features, whether it cares about the position in the image or not. All of this is optimized to be encoded in those voxel embedding vectors. Note, only the voxel embedding is unique per voxel. The other model components are shared. This allows our model to be trained on much more data, as the shared components can be trained on all voxels, both within a specific brain, but also across different brains through this shared voxel embedding space. Altogether, this gives us a very strong encoder, as Roman will show you now.
Thank you, Navve. Let's talk about evaluating our encoder. A subject sees an image, and we measure the corresponding fMRI response. We can now use our encoder to predict the fMRI signal. We evaluate our encoder using multiple quantitative metrics, among them correlation and retrieval, where it consistently outperforms previous approaches. We provide detailed evaluation in the paper. Let me show you one interesting visual evaluation. Given a measured fMRI response, can we retrieve the image the subject actually saw? The answer is yes. We can use our encoder to find the image that best matches the measured fMRI. Here, we see the top 5 retrieved images out of a set of 1,000 images. The first image is indeed the correct one, but what's also interesting is that the other 4 images are also very similar semantically. We can see this pattern across many examples, and here are a few. In the top row, we can see that all the images are images of airplanes. Overall, we get 95% top 1 accuracy of retrieving the correct image out of a set of 1,000 images. We trained our model to learn an embedding for every voxel. Can we understand something about what we learned? Are the embeddings actually meaningful? Let's zoom in into the voxel embedding space. Each point corresponds to a specific voxel. We can observe that it's not uniformly distributed, and there are distinct clusters that are formed. But what does it mean for a cluster to emerge in a voxel embedding space? Remember, each voxel was optimized in its own embedding separately to encode what it cares about in images. Yet different voxels in different brains learned close voxel embeddings. This cannot be accidental. It means that all the voxels in the cluster share something which is functionally meaningful. We will map the resulting functional clusters back to the brain. Here are 4 example clusters for one hemisphere of a single subject. Notice that each cluster is specially localized. Now let's look at the second subject. Because the embedding is shared across subjects, we can perform co-clustering and find the corresponding functional regions across people. The model has no anatomical information or voxel location. This organization is learned entirely from the data. So what does each cluster actually represent? Let's look at images that highly activate each cluster. Here, for the orange cluster, we can see in the top row the images that highly activate this cluster for subject 1, and at the bottom row for subject 2. Even though each subject saw completely different images, the semantic categories emerge. Here is another cluster: sports, text, and faces. Can we learn more about known regions? Let's focus on the PPA, a well-known region for places representations. We apply the same clustering procedure within the PPA without any supervision. 2 distinct functional clusters emerge. One is selective for indoor scenes, the other one is selective for outdoor scenes. Let's summarize. We presented the universal image to fMRI brain encoder. It combines information from multiple brains, datasets, and machines without any shared data. It facilitates the discovery of shared functional information across different individuals. Something I haven't covered but is important is that we have an efficient adaptation to new subjects with as little as 100 subject-specific examples. If you want to know more and would like to discuss the paper, please come to our poster number 2, session F tomorrow, or look at our CCN proceedings paper. Thank you.
Thank you, Navve. Thank you, Roman. And is there any question from the audience? Okay, I have a question for you guys. Yeah, because you didn't mention here, but I noticed that in your abstract, you mentioned that there's a observed lower performance in the higher order or the semantic related vision areas. And have you considered using a multi-model vision language backbone instead of vision-centric backbone? Because in this case, I think it can.
Sorry, generally I think low visual cortex, the SNR is higher a lot of time, and therefore the performance is better. We did try both. The backbone we are using is Dino, but we also tried Clip, that it's kind of textual, and it's worked I think almost
more or less the same.
More or less the same. So we didn't see any improvement by using something that is more semantic.
So you think it's more because of the noise instead of
it's only inherent to the data.
Maybe.
Itself.
Or more differences between different subjects also might influence, because if you have more variety in the higher level, but we don't know exactly yet.
I see. Thank you so much. And yeah, guys, if you're interested, please check out their posters and their preparing. Yeah. Our next speaker is Jon Gauthier, who will present neural footprints, variance-based methods, systematically meets high-level representations. Please join me in welcoming Jon.
All right, hi everyone. This is a collaboration with Tyler Brooke Wilson at Yale. And today I want to suggest that some of the representations that we most care about finding in the brain may be systematically invisible in naturalistic experiments. We will explore this idea by thinking about vision. So in vision, as in other domains of perception, we transform a massively high-dimensional sensory input, this array of pixels over here, through a series of computations into a very abstract representation. And abstract representations are what enable many feats of visual intelligence, from forming expectations about what might happen next in this scene, to forming plans about how to intervene in the scene successfully to achieve our goals. And 2 issues arise when we think about how to discover the neural correlates of these representations, these 2 levels, when thinking about this from a computational perspective. So the first we call a footprint asymmetry, and that is that the neural signal is going to be dominated by this massive high-dimensional sensory data relative to the abstract data. The second is that these 2 levels of representation are deeply confounded in practice. These ideas are not new to anyone in the audience, I'm sure, but the contribution of this talk is to demonstrate that in actual practice, in a simulation experiment that I'll show you next, these two levels are so confounded and so asymmetric that we cannot identify abstract representations at all. So here's our simulated environment. We're going to study a model system operating in this simulated environment. And that model system perceives objects in the foreground, moving from either left to right or right to left. And the job of our model system, which we trained to perform this task, is to map from an array of frame representations, these pixel arrays, into an abstract physical representation of the scene. And that abstract representation is sufficient, we demonstrate in the paper that it is, to predict future frames of the same scene. So we get a prediction like this for the current scene. So we have this model that we know is accomplishing an interesting task of visual perception, and we're going to study it, in particular its brain state. We're playing the game of cognitive neuroscience here on the simulated brain state of this model. So me and my co-author Tyler will look at this brain state from the outside, treating it as a black box. And we're playing the best possible case game of cognitive neuroscience in which we actually have access to the ground truth representations. We know for every scene the inputs and the outputs of this model at the low and the high level. So our question is, can we recover those two levels, or traces of those two levels in the brain representation? So this simulated neural data is a simple linear projection of the inputs and outputs of the model into a flat array of neurons. And this flat array is what we're studying, asking whether we can recover traces of those two levels of representation. Okay, so we'll first approach this simulated brain data with a basic unsupervised analysis. We're going to think about our domain a little bit and notice that a major salient dimension of variation across scenes is that, well, sometimes objects move from left to right, and sometimes they move from right to left. And this should be something that's represented, obviously, in the model's representations and in its brain representations. So let's do a simple PCA, principal components analysis, on the neural data that's tokened by this model in response to each scene. So here's a PC plot. Each point here will be a single brain activation in response to a single scene. And we're coloring the points based on whether the relevant scene has objects moving left or right. And the answer, as you may expect, is that motion is not encoded anywhere in these first two principal components. What they encode instead are very coarse sensory facts. Here from left to right, we have something like the brightness of a scene, and from top to bottom, something like the zoom of the camera. These are distinctions that have massive consequences for all the pixels in the scene. Looking further across the spectrum, we can apply a decoding approach and ask, can we decode facts about motion anywhere in higher order principal components? And the answer is eventually yes. At higher order principal components that explain less variance in the brain data, you can reconstruct the direction of an object's motion. So this first just demonstrates the basic idea that there's an asymmetry of footprint, right? We have to look far out in the spectrum in order to find some trace of putative high-level representation. But we're not finished yet, because while we could be tracking something abstract here, we also could be tracking some neural trace of a sensory confound of motion. And so to control for that, we'll think about this space of total variance of brain data. And we'll partition it into two parts. There's the part that could be explained from pixels alone. This is data that could possibly be a sensory representation. And then there's a residual space not explained by pixel data. And we're interested really in this residual space, controlling for the left side. Can we still reconstruct something about objects' motion? If we can't do that, that indicates that the original positive result was simply a result of sensory confounds in the neural data. And that's actually the case, unfortunately. After controlling for possible sensory confounds, at no level can we decode motion facts from the neural data. So this illustrates our second point of confounding. To the extent that we have any abstract representations visible in the neural data, they're so deeply confounded in practice in these modeled representations that after controlling for sensory facts, there's nothing left to explain. Okay, but we have an easier job here, actually. We're operating with the ground truth representations that we know are present in the model and its brain representations. So we applied a brain encoding approach, a linear regression, trying to map from either the pixel representation of a scene or the model's physical outputs onto individual neuron activations. And we see that pixels predict neuron activations quite well across scenes, whereas physical information barely does at all. So again, just a very simple illustration of the asymmetry to begin with, that sensory data occupy the majority of explainable variance in the brain data. While this provision looks positive, that we can explain some brain data as a function of that abstract physical quantity, again, recall that our model in practice has confounded sensory and abstract representations. So we'll control for that again by thinking about different components of variance in this brain data. Now, we're interested really in the brain data that can be explained uniquely by abstract representations. And we want to control for this center area that could plausibly be explained by either sensory or abstract representations. What I showed you before, this result was really a measure of this circle, and we care about controlling for that center part again. So after controlling for that center part, we can calculate the unique variance of the brain data explainable by physics relative to the original sensory result. And the answer is approximately 0. The answer lies within a null distribution that assumes there's a random relationship between a model's representation of the abstract features of a scene and its brain activations. So we played the game on the easiest mode possible. We have access again to the ground truth representations at the high level. We know how they're coded, the format of the code as well. But we found that those most critical representations that are relevant for, say, forming expectations about what might come next in a scene are the ones that are least visible to our methods. And that happened for 2 reasons, right? We have the asymmetry representation in the first place and their footprints. And second, this empirical fact that these 2 levels of representation are deeply confounded in the model. This matters not just for identifying representations, but also for doing things like model comparison. So we think it's problematic if you're looking to select models based on their explained variance in neural data as a good proxy for their computational similarity to what happens in the brain. Following what we've shown here, if 2 models differ in their explained variance of brain data, it's almost certainly the case that is because of their sensory representations and not because of the way that they code abstract information about an input. So this is a very summary overview, of course. I'd be happy to debate and think about variations of the simulation paradigm tomorrow at our poster F1. And for now, thank you for your attention.
Thank you, John. And I think we'll take 1 to 2 questions from the audience.
Isn't the brain slightly easier because it partitions the visual information from the abstract in different areas of the brain?
So, okay, so it's a question about sort of like functional specialization of the brain. So what we'd like to do next, basically what we'd like to establish here is a paradigm where we can be specific about the assumptions we need to make in order to recover abstract representations. And it's possible, we need to bake this in 2 parts. First of all, the neural simulation itself has no functional specialization. And secondly, we might bake that into the actual analysis. So assume functional specialization, you have spatial localization of some kind of level of representation, and show that this works. So I'd like to try 2 things. First of all, assume no spatial localization of function, but bake it into our methods. Do we get false positives? I would like to know that first. The second is, if we do have functional specialization across the brain, can we find false negatives, or is there a space we can stand in our analysis where we find just the true positives and the true negatives, and no false negatives or false positives? So I can talk more about that later at the poster if you'd like, but what I'm excited about here is that we can have a paradigm where we can explicitly state what kinds of assumptions are necessary for us to succeed in this game of cognitive neuroscience. Thank you.
Thank you so much. And so let's welcome the final speaker in this session, Jon Vastola, who will present implications on hierarchical Markov models of behavior on irreversibility, predictability, and dimensionality. Welcome, John.
All right, hello everybody. I'm Jon Vastola. I'm a postdoc at Harvard. When I was in high school, I did a little amateur ethology project. For reasons that are kind of unclear to me, in my grandma's neighborhood, there was this group of peacocks that lived there and sort of wandered around all the time. So I thought it'd be fun for my biology class to follow them around for a day, record where they were roughly every 5 or 10 minutes. So I did this for an entire day, recording the little notebook as I followed them where they were. And not only was it extremely boring, but also I feel like I didn't really learn that much about the peacock's behavior. I did say that I saw a peacock see itself in the mirror and then like get confused and attack itself, so that was interesting, but maybe that's not like the most interesting scientific conclusion. But the sort of great ethologists of the past, people like Konrad Lorenz and Nico Tinbergen, kind of had to do similar things. They had to go out into the wilderness and like rough it and track animal behavior. And it's really difficult to do this. But fortunately, perhaps, times have changed. We have kind of an unprecedented ability now to track behavior at large scale in an automated way. There's been a lot of advancement in pose tracking. There's some papers that are relevant for that. There's been advancement in taking these like movies of behavior and segmenting them into chunks and saying in this sequence of frames, the animal's kind of doing like a little walk. In this sequence of frames, the animal's doing a turn. In this sequence of frames, the animal's like rearing up or grooming itself or something like this. And people have even gone beyond that to construct models that say how often you transition between these different high-level behavioral syllables. So I'm not going to talk about any of those individual things, which a lot of people have put in a lot of technical work to get right. What I am going to talk about is, now that we have this thing and it kind of works, what do we learn about behavior? So I just want to say briefly a little bit more about the kind of model that you're coming up with. The idea is that you have different behavioral states. So for example, you could have three things like explore, rear up, or groom. And you can imagine in a movie you sort of transition between them empirically at some rate. You can collect these rates into a matrix, and the sort of assumption of the Markov model is that the way your distribution of behaviors changes over time, the probability that you're rearing or grooming or exploring is governed by this transition matrix. So I'm going to call that H. And if you're sort of a formal mathy person like me, you can collect this into what's called a master equation. So it's this linear equation which tells you how the probability that you do any particular behavior changes over time. The key assumption underlying this model is pretty strong. The assumption is that whatever I'm doing now completely determines what I'm going to do next. Or in other words, there's no kind of higher order memory. It used to be the case that work would explore non-Markovian structure and behavior. And they would show that this kind of assumption is like obviously wrong. But what's extremely interesting within the past few years is that it's been shown that if you account for latent states, perhaps by taking into account structure in simultaneous neural recordings, you can actually get Markov models that fit. So yeah, the key question I want to ask and present is, if you have this Markov model, and if it fits, and if we take it extremely seriously, what does it really tell us about the large-scale structure of behavior? And I claim that you can use this model to come up with model-based answers to interesting fundamental questions about the structure of behavior. So I'm going to talk about three things extremely briefly. The sort of sequence-like or irreversible character behavior, that's the first thing. The second thing is what we mean by the dimensionality behavior. And the third thing is sort of talking about some consequences of hierarchical structure and behavior. The fact that you have sort of sub-second ongoing behavioral states, but also latent states like whether or not you're hungry. But if you want full details, which I can't treat here, you can check out our proceedings paper. So the first thing is that if you watch a movie of behavior, is it going to look different if you play it forwards versus backwards? For a long enough movie, it will look different because at the end of the movie, the animal will be dead forwards. But for shorter time scales, the question can be a little bit more interesting. In other words, does the animal do A, B, C, D, A, B, C, D, A, B, C, D, or does it kind of go A to B or B to A or whatever? And it turns out you can formalize this by comparing your Markov model to its time reverse. There's different ways to do this mathematically, but one of them, which is kind of interesting, canonical, is called the entropy production rate, which has been studied in physics quite a bit. It's sort of like considering the Kale divergence between the sort of forward transition and the backward transition model. So if you do this, you get kind of results that make sense for a toy model. For example, if you have sort of like a ring of states, A, B, C, D, A, B, C, D, A, B, C, you tend to do sort of in a stereotyped order. You're going to find that the irreversibility grows the more you tend to go in one direction and not the other direction. And in real data, you can also quantify this irreversibility. for example, across different behavioral states. And you can find things like when a mouse is being more social, its behavior is a bit more sequence-like or irreversible or directed than if it's like randomly exploring. So I think that's kind of interesting. The second thing I want to talk about is briefly the dimensionality behavior, which is like kind of a fundamental theoretical question. I'm not going to talk about chunking behaviors, but rather I'm going to talk about chunking structure in the transition matrix or in your probability distribution. And one way to think about it is like this. For simplicity, imagine that there's only 3 behavioral states. It turns out mathematically you can show under certain assumptions that your probability of doing any behavior over time has 3 parts. One is sort of like pi, the thing you're doing at equilibrium. And then there's 2 other parts that represent transient contributions. And these can decay at different rates. So you can plot your probability vector as a function of time sort of on this axis of like the extent to which the V1 eigenvector of the transition matrix is still there, and the extent to which the V2 component of the transition matrix is also there. So there's the dot. It tells you how far you are from equilibrium. Assuming that there is some separation of time scales, after some time, one of the components is going to die. And so in that sense, you can say that this p vector is like lower dimensional than it was originally. And then sort of on a long enough time scale, since you just go to equilibrium, it becomes even more trivial. You don't need any basis vectors apart from your steady state distribution. So you can think about this more generally. In general, something called the Perron-Frobenius theorem allows you to like deconstruct this p vector in terms of the eigenvectors and eigenvalues of the transition matrix. And what I think is interesting is that these eigenvalues tell you something about the fundamental timescales of behavior, and the eigenvectors tell you something about the characteristic ways that behavior changes over time. So one way to define dimensionality I think is interesting is in terms of the number of basis vectors you need to specify how close you are to equilibrium. And so this sort of has reasonable properties, like very unstructured behavior is construed as high dimensional, and highly structured behavior like ABCD is construed as a very low dimensional. And real behavior is sort of like somewhere in between those 2 things, which kind of makes sense to me. There's sort of another way you can do this based on mutual information, but it ends up getting you to kind of the same place. So conceptually it's a bit different, but quantitatively it doesn't look so distinct. And again, real data is somewhere between sort of maximally unstructured and maximally structured. The very last thing I want to say briefly is that the Markov models that fit nowadays have a sort of subtle notion of state, which doesn't just include the thing you're doing right now, like grooming or walking or turning, but also includes some notion of latent state, whether I'm being social, whether I'm exploring, or whether I'm hungry, something like this. And it turns out models with this kind of structure exhibit sort of interesting spectral structure when you look at their eigenvalues and eigenvectors. It turns out there's sort of fast eigenvalues and eigenvectors that correspond to the transitions between ongoing behaviors. And then there's sort of like slow timescales which come from the switches between different latent states. So again, like whether I'm transitioning between being social or being hungry or grooming myself or something like this. So I think this is interesting. This is a bit technical, so I can't say too much more about it here. The sort of high-level take-home message, which I just want to convince you of, is that now that we have these models that can fit pretty well, I think there's interesting theory that we can draw on to understand the structure of behavior. I gave you some examples, but I think there's probably a lot more we could do, and I think this is worth thinking about more seriously. Thank you.
Thank you, Jon. And is there any questions from the audience?
Hi there. Yeah, cool talk. For these fast transitions, I'm wondering to what extent do you think this might be due to misspecification? Like, is it that there's quick transitions between hidden states that correspond to similar kinds of actions where because you're discretizing the action space, it has to carve things up more finely than it needs to?
Sorry, so just to be clear, I couldn't hear that well, but the question was about...
what's the structure of these transitions? Like when you find these kind of short-lived, kind of fast transitions between states, if I understood correctly, maybe I
Sure. So, well, I think the similarity of different actions plays into it. I think the main thing, and again, I didn't do this work, I'm drawing on this work, is looking at simultaneous neural recordings. So if you watch neural recordings, then you can decode from those recordings. You can identify and decode certain latent states which are consistent on a sort of longer time scale than the moment-to-moment behavior. So things like maybe tens of seconds or longer. I think it's that which determines the slow structure. I think it's important to point that out because the specific thing you could be doing moment-to-moment could be the same between different latent states. What's important is that whether I'm hungry or social or whatever changes the rate at which I transition between the states.
Yeah, it's just not always clear whether or not those neural states are also discrete or whether or not they're continuous.
Sorry, I see. This existing work claims that it fits well to treat them as discrete, but you know, who knows? I think it's a good question.
Okay, thank you, John. And with that, we conclude the theory and methods session. Let's thank all of our speakers again for a great set of talks. And thank you all for being here and enjoy the rest of the conference. Thank you.